Bismillahirrahmanirrahim. Other levels to regulate the gene expression of the messenger RNA processing. This is our lecture 5 in respect to the regulation of gene expression. First of all is the alternative splicing. As splicing method, by means of splicing method, exons they joined while the removal of intron take place in the messenger RNA. Splicing is the eukaryotic phenomena, and in this case the joining of exons and the removal of the intron. By means of alternative splicing, there is the joining of different exons. It is not necessary that in joining of the exons, the whole exons they are joined, but these exons they are the alternative joining of these exons lead to the isoforms of different proteins according to the tissue differentiation. These joining, for example, this one is the protein 1 and protein 2. Here the joining of exon 1, 2, 4 and 5 skipping this exon, and here is the 1, 2, 3, 5 along with the skipping of this exon. Here is the skipping of this axon, and here is the skipping of this axon. They form the different isoforms of the protein, and these proteins they are functional according to the cellular differentiation or tissue differentiation. So alternative splicing also take part in the gene regulation. Another way by means of alternative splicing take place: exon 1, 2, 3, 4, 5. 1, 2, 3, 4, 5 the protein is formed after the translation, while here there is a skipping of this exon, thus the isoform of protein is formed. Different isoforms are formed according to the tissue differentiation by means of these alternative splicing. Now the polyadenylation take place when polyadenylation is present at the 3' end of all the eukaryotic of almost all the eukaryotic messenger RNA, and this 3' end they may be uh, editing mean may be shorter in length or longer, and if they are cleavage or the editing take place at the poly tail, then it may be the coding region may be greater, longer as in this case, and or maybe it will be a shorter and longer poly tail. So depends upon the poly tail. It also give rise to different isoforms of proteins. Polyadenylation is another way for the regulation of gene expression. The polyadenylation may take place at the coding region or may take place at the poly tail, and this is the, for example, in the poly tail cleavage, a messenger RNA that contains the poly A tail, it may be proximal cleavage site or distal cleavage site, and it give rise to the short poly A tail, or may give rise to this one. Similarly, different 3' UTR lengths they are produced, and here the this one the cleavage take place at this or may take place at this, and alternative C terminal sequence from use of intronic stop codon, and different by means of this they also give rise to the different protein isoforms. So by increasing or decreasing the length of poly A tail and along with the coding region, different isoforms of protein they are formed. Messenger RNA editing, it is also utilized for the regulation of gene expression, as this editing they may give rise to the premature termination of uh, messenger RNA, or premature termination of transcription. For example, the CAA, it is the codon uh, uh, present here, and this codon, it is the codon for glutamine in the liver. This code for glutamine, and ApoB100 is produced. Apoproteins, they are produced in lipoproteins, and they are used to transport the lipids from one place. These are the vehicles, and these apoproteins, they are present in the lipoproteins. In liver, B100 apoprotein is produced as CAA. It's code for glutamine. But what happens in intestine? B48 apoprotein is produced because the messenger RNA, it is edited, and it is truncated proteins. It is edited in such a way that truncated protein is produced. This CAA is edited at C is replaced by U, and now UAA is the stop codon, and in the intestine, the stop codon, the messenger RNA. Now here is the formation of the stop codon, and this is the only length of the messenger RNA, and only 48% messenger RNA it is translated. So in intestine, the 48% messenger RNA is translated, truncated protein is produced, which is known as the ApoB48. ApoB48, it is necessary for the functioning of chylomicrons, but in liver, ApoB100, the whole, there is no editing, and the whole messenger RNA, it is translated. By means of messenger RNA editing, different forms of proteins, they are produced. Messenger RNA stability. Messenger RNA stability is also necessary for the regulation of gene expression. How long this messenger RNA it is exposed for the translation, it depends upon the condition of the cell from where this messenger RNA is produced. For example, in erythroblast, transferrin, it is the plasma protein, and it uses the transferrin receptors in erythroblast. This transferrin, it is the iron carrying protein in plasma, and these, when joined with their receptors, it give their iron to the erythroblastic cells. What happened? The transferrin receptors, the messenger RNA of the receptors, they are basically the proteins. The messenger RNA of transferrin receptors, they contain the stem and loop structures at the 3' UTR region. These 3' end, the stem and loop structure, these are these are known as the iron, and these structures, they bind with the iron regulatory proteins. Whenever they bind with these iron regulatory proteins, they are they are stabilized. This messenger RNA is stabilized and produce the receptors. When there is a low concentration of iron in the erythroblast, what happens? These regulatory proteins, they bind with the iron response elements and stabilize this messenger RNA, and now this messenger RNA is now it is stabilized to produce the transferrin receptor. So more and more iron will be entered into the cytosol. But when there is a high concentration of iron present in the erythroblast, then what happens? These regulatory proteins, they are detached from these response elements present at the 3' side, and after detaching, they destabilize this messenger RNA, and no transfer, because they degenerate this messenger RNA, so no messenger RNA is available for receptor formation, and these receptors now, there is no more receptors. So by stabilizing or destabilizing these messenger RNA, they can control the stability of the messenger RNA, and then the entrance of iron into the cell. Another protein known as the ferritin. Ferritin contains it is the storage protein in the cell, and it contains this structure at the 5' end. And when there is similarly it is stabilized and destabilized when there is a high concentration of iron in the cell. It means there must be a ferritin to store this iron, and there is a opposite action. This protein, it is detached from this structure present at the 5' end of the ferritin messenger RNA, stabilize ferritin messenger RNA so as to produce the ferritin. But when there is low concentration of iron in the cell, then what happen? It means there is no purpose for the storage of the iron in the cell. So these proteins, they bind with this stem and loop structure present at the 5' end and destabilize the messenger RNA. RNA interference. It is a phenomena by which the messenger RNA can express itself, or can be degenerate, or can be its translation be halted. RNA interference is the phenomena by which the micro or it is the silencing, gene silencing, and here the micro RNA can be produced, or a small interference RNA can be produced. Micro RNA, they are produced in the nucleus as a double-stranded, and by means of action of dicer. It is now dicer is present in the cytosol, while the dorsal is present in the nucleus, and by the action of dorsal and dicer, this double-stranded now becomes the single-stranded and short length. Now one of the strand of this micro RNA or small interference RNA, it becomes the guiding strand for the protein risk, that is the RNA induced silencing complex. And this complex, it by means of other proteins like agoproteins, argonaut, four proteins of the argonaut, one of the four protein of argonaut, it may degenerate the messenger RNA and then halt its expression. Here the double-stranded RNA that has been produced, and after the action of dorsha, and it will be entered into cytosol, from where dicer it cleave at different region. Micro RNA produced within the body, while the small interference RNA, it has been given from outside sources, and one of the strand is the guide strand from which the RNA induced silencing complex it join, and this complex may after when it join with messenger RNA, this is the at the specific level on the messenger RNA, it cleave the messenger RNA, or may it halt its translation of the messenger RNA. This RNA interference therapy is used for many diseases. First of all, it was used in the age-related macular degeneration, which lead to the blindness in all over the world. This disease is due to the overproduction of vascular endothelial growth factor, and for the silencing of the messenger RNA of vascular endothelial growth factor, this factor can't be produced, and thus this disease can be prevented. Due to the overproduction of vascular endothelial growth factor, there is a sprouting of the excess blood vessels behind the retina, and due to which the blood vessels leak, cloudy, and destroying of the vision. This is also known as the wet macular degeneration. Small interference RNA It is used as a short double-stranded RNA, and it will destroy the messenger RNA of the vascular endothelial growth factor, and this degeneration and degraded this messenger RNA and prevent this disease. Um, One of the small interference RNA, it can destroy hundreds of messenger RNA, thus the suppression of thousands of vascular endothelial growth factor proteins, and thus prevent the angiogenesis in the retina. Messenger RNA translation. The expression can also be stopped at the translational process, and here one of the translational enzyme, which is known as the eukaryotic initiation factor 2, when it is phosphorylated, then it will be inhibit, and this inhibition, it is due to the presence of the kinases, and these kinases, they will be activated due to the amino acid starvation, heme deficiency in erythroblast, signaling viral infection or accumulation of misfolding proteins. All of these reasons, they can activate the kinases and inhibit the eukaryotic initiation factor 2 by means of phosphorylation. The eukaryotic, here is the phosphorylation of eukaryotic initiation factor 2. In the eukaryotic initiation factor 2, when it is phosphorylated, then it means there is no translation. However, the translation occurs when there is a phosphorylation. Access of DNA is another way by means of acetylation to form the euchromatin, and there is the phenomena by which the acetylation take place, and the access of transcription factor to their respective DNA nucleotides take place. But if these are in condensed form, then there is no access and no transcriptional no transcription take place. Here the acetylation, which convert the, this is known as the chromatin remodeling. Here the chromatin heterochromatin is being converted into the euchromatin, and again this is the reversible phenomena. Again it will be converted to the heterochromatin. This chromatin now, it has great access to their transcriptional factors. Now these transcriptional factors, they are attached to the nucleotide sequence, but there is no access towards the nucleotide sequence. This is another way for regulation of gene expression in eukaryotes. Methylated histones, when histones, they are methylated, methylation is the post-translational modification, and when they are methylated, then there is a less access uh, towards the transcriptional factors as they are in condensed form. The methylation gives the condensed form of the chromatin, while the less methylation, it will give the, these are the methylated molecules, and it will give the less access, while the less methylation, it will give the more access towards the transcription factor as this chromatin is less dense. Here is the histones H3, as there are the different types of histone molecules, protein molecules H1, just like H3 protein. This is, it is methylated by the histone methyl transferase enzyme, and maybe it is demethylated. DNA recombination takes place to form the different immunoglobulins, and these immunoglobulins in the variable region. This also gives the regulation of gene expression as the variable region, diversity region, and the joining region. They form to combine the various types of the immunoglobulins. Thus, the 10 raised to power 9 to 11 immunoglobulins can be formed by a single lymphocyte. Transposons, these are the DNA segment of DNA that can be joined to other chromosomes by means of transposases. These are the enzymes. These transposons, they may be cleaved and joined to the other segment, directly joining take place, or may they are copied. For example, they are first of all be copied, and this copy is joined to the other chromosomal or DNA sequence. So maybe directly or maybe indirectly, and this gave another way for the regulation of gene expression. Now there is a end for the chapter gene expression, regulation of gene expression both in eukaryotes as well as in prokaryotes. Thank you very much.